Introduction to the immune system. Innate immunity is natural immunity, so this is the ability to resist infection by means of normally present body functions. It's nonspecific, and the response is the same for all foreign substances. So examples of innate immunity would be like your skin. So naturally, every person has skin on the surface of their body, and that skin has a low pH, high salt concentration that deters the growth of many microorganisms. You typically don't see moldy people walking around because molds don't really like to grow in the presence of that low pH or that acidic environment or high salt concentration. So, skin isn't specific. It keeps out pretty much every type of microorganism, whether it's a parasite, a mold, a bacterium, a virus. It's nonspecific. It works to keep out everything. And the response is the same for all foreign substances. And the skin is just one of many examples that we're going to talk about as we get into more detail about innate immunity, but it's nonspecific. Adaptive immunity is specific. It's a type of resistance that's characterized by specificity for each individual antigen and the ability to remember a prior exposure. So an antigen is a component of a cell that your immune system has the ability to recognize. And there are self-antigens, or components of the cell that belong to our cells, and those are okay to stay in the body. But then there's foreign antigens, and foreign antigens we detect and we want to eliminate or remove from the body. The adaptive immune response and adaptive immunity has to do with that specific recognition of a certain type of antigen. So your immune system has the ability to recognize very specific components and determine that it's non-self, and then develop memory against it. So if you ever encounter that exact same antigen again in the future, it remembered that previous exposure and mounts an overwhelming and very fast and rapid response so that it can clear that antigen even quicker upon second exposure. Again, we'll talk more about adaptive immunity when we get to that portion of this course. Cells of the immune system. So your immune system basically relies on different types of cells, and those cells are going to recognize and respond to foreign substances, cancer cells, any abnormal substance that comes in the body, all that type of stuff. The cells that the immune system uses are the white blood cells, also known as leukocytes. So in your bloodstream, you have erythrocytes, which are the red blood cells, and you have leukocytes, which are the white blood cells. Of the leukocytes, there's granulocytes, agranulocytes, and then there's a whole bunch of other cells that aren't categorized as one of these two types. The granulocytes have granules, and those granules are visible through staining procedures. And the granules contain certain types of chemicals that mediate different types of responses. Neutrophils, eosinophils, and basophils are examples of granulocytes. Agranulocytes do not have these obvious granules. And if they do have granule-like components to the cell, they are very tiny and they don't contain the necessary chemicals to be classified as granulocytes. So agranulocytes include monocytes and lymphocytes. So looking at this, you can see kind of the progression of how a cell would start out as a stem cell and then differentiate into the different types of cells of the immune system. So you start with this hemocytoblast, which can turn into a myoblast, and this myeloblast is more differentiated than a hemoblast. Then that can turn into a progranulocyte, and progranulocytes will further differentiate into basophils, eosinophils, and neutrophils. Hemocytoblast can also turn into a lymphoblast, and a lymphoblast can become a lymphocyte. Can also become a monoblast, and a monoblast can become a monocyte. So neutrophils, this is what neutrophils look like in the blood. So they're relatively large cells compared to your red blood cells, But the key thing here is the multi-lobed nucleus. So the nucleus is kind of skinny and you can see multiple lobes to the nucleus. Neutrophils are the most populous or most populated white blood cells circulating in the bloodstream. About 50 to 75% of circulating white blood cells under normal conditions would be neutrophils. The 2 to 5 lobe nucleus give it its nickname of being segmented or segs. They can also be called polymorphonuclear neutrophils or PMNs. So sometimes when you report data or take down results or even get results from some of the analyzers, you can see segs or PMNs. When we do microbiology reports to look for the presence of pus, we look for PMNs, which is referring to the neutrophils, because neutrophils would be pretty plentiful in pus samples. So segs, PMNs, they all refer to neutrophils. It's very important that you recognize those abbreviations because a lot of times texts will use those terms interchangeably, so it's important to make note of that. Phagocytosis to clear foreign substances, including bacterial cells. So that's neutrophils' primary goal is to eat things. Phagocytosis, cellular eating, so it will come up to a foreign substance, engulf it, and then it has granules inside that can digest the components that it just consumed. Neutrophils are also major mediators of inflammation, and we'll go through that whole inflammatory response in future lecture. Eosinophils. 1 to 3% of circulating white blood cells are eosinophils under normal conditions. Increases in eosinophils are associated with allergies or responses to certain parasitic infections. Eosinophils have bilobed nucleus, so you typically see 2 portions of the nucleus, and the granules typically stain red or orange. They regulate mast cell function. Basophils, less than 1% of circulating white blood cells under normal conditions. The granules stain blue or purple. They're very large granules, and this isn't the best picture of a basophil, but you'll see very large and dense granules, and you will see that like purplish, bluish color. They often hide the nucleus of the cell because the granules are so large. Basophils help mediate allergic reactions, and they play a role in anticoagulation. Moving on to some agranulocytes, starting with the lymphocytes. So lymphocytes are 20 to 40% of the circulating white blood cells under normal conditions. There's B cells, T cells, and natural killer cells, and they play a role in adaptive immunity. So if we look at this myeloid stem cell, myeloblast becomes the granulocytes, and those are the different types of granulocytes we just talked about. A lymphoblast has the ability to become different types of lymphocytes. So the B lymphocyte, T lymphocyte, or natural killer cell. Natural killer cells we'll talk about more in the future, but just because as a preliminary definition, a natural killer cell will attack and kill cells that appear to be abnormal. This includes cells that are infected with viruses and cancer cells. Monocytes. So monocytes are one of the largest white blood cells. They're pretty massive compared to a red blood cell, and you typically see a large nuclear pattern, so a U-shaped nucleus. You won't see any obviously stained granules in the cytoplasm, but this nucleus will take up the majority of the inside of the cell. 4 to 10% of circulating white blood cells under normal conditions are monocytes, and they remain in circulation only for a short time until they migrate to the tissues to become macrophages. So once they leave the bloodstream, they'll become macrophages. Macrophages play a role in initiating and regulating innate and adaptive immune responses. In the innate immune response, they play a role in microbial killing, anti-tumor activity, intracellular parasite eradication, phagocytosis, the secretion of cell mediators, and they're the basis for tattoos. Macrophages are giant eaters, so they like to consume foreign substances. In the case of a tattoo, when the macrophage sucks up the dye that's been injected into your skin, that dye can't be degraded. It can't be broken down by what's inside of the macrophage. So that dye remains stable inside of your macrophages, and if a macrophage dies, a neighboring macrophage will consume the dye that spills out of the dead cell. So that's why tattoos kind of stay in place. Mast cells. They resemble basophils but come from a different lineage. They're in tissues, so you wouldn't typically see these in circulation. They play a role in mediating allergic reactions. They can also function as antigen-presenting cells and can enhance and suppress the adaptive immune response. Dendritic cells are extensions that make them resemble nerve cell dendrites. So they have these projections that stick out. They're the most effective antigen-presenting cell in the body and the most potent pathogenic phagocytic cell. So I've mentioned antigen-presenting cells a couple times. We will get more into detail about what an antigen-presenting cell is, but basically a dendritic cell will eat a bacterial cell, for example. It'll chop it up and then it will gather a specific antigen that's part of that bacterial cell, and it will put it on the surface, and it will present that to your immune system saying, this is what's inside the body, this is what we just ate, pay attention to this. And that's how your body knows that you've been infected with a certain type of pathogen. So that's one means for activating that adaptive immune response, and we'll talk more detail about that as we go through the next few lectures. Organs of the immune system. So there are primary and secondary lymphoid organs. The primary lymphoid organs are different from secondary in that the primary lymphoid organs is where your immune cells mature. So the immune cells, a lot of times they come from the bone marrow. They're going to be stem cells in the bone marrow. And then certain immune cells will then have to travel to the thymus to continue their maturation process. So those are the two major primary lymphoid organs that you're going to see, bone marrow and the thymus. So for bone marrow, it's one of the largest tissues in the body. Bone marrow is found inside of your bones. It fills the core of all long, flat bones, and it's the main source of hematopoietic stem cells, which develop into erythrocytes, granulocytes, monocytes, platelets, and lymphocytes. The thymus is a small, flat, bilobed organ that's found in the thorax. So here on the right, you can see a picture of the location of the thymus. It is the location for the maturation of T lymphocytes, or T cells. Secondary lymphoid organs function as potential sites for contact with foreign antigens. It's kind of where I like to say that the white blood cells hang out and hope to be coming in contact with antigen. It increases the probability of an immune response. It includes the spleen, lymph nodes, mucosal associated lymphoid tissue, or MALT, and cutaneous associated lymphoid tissue, or CALT. It is important to know what MALT and CALT stand for, and you should make note of the full written out words of what those stand for. So the spleen has two main types of tissues. There's red pulp and white pulp. The red pulp makes up more than 1/2 of the total volume, and that's where red blood cells go to die. So if you have circulating red blood cells that are getting old or got damaged, that's where they get filtered out of the bloodstream. White pulp comprises about 20% of the total weight of the spleen, and it contains lymphoid tissue arranged around arterioles in a periarterial lymphoid sheath containing T cells. Basically, it's a region that's lighter in color. It contains some T cells, and it's a dense region of T cells so that when blood is rushed through the spleen, it comes in contact with these T cells, and the T cells will snag and bind to any foreign antigen that it happens to recognize. And that can stimulate the progression of a full adaptive immune response. Lymph nodes are central collecting points for lymph fluid. Lymphocytes and any foreign antigens enter the nodes by different lymphatic vessels, or by afferent lymphatic vessels. There's secondary follicles that consist of antigen-stimulating proliferating B cells. The location where transformation of B cells takes place form plasma cells and memory cells when exposed to an antigen. So that whole progression of B cell activation will be discussed once we get into the adaptive immune response. MALT and CALT, as I mentioned, mucosa-associated lymphoid tissue. It's found on mucosal surfaces of the GI tract, respiratory, and urogenital tracts. It's a main port of entry for foreign antigens. There are numerous macrophages and lymphocytes present. Cutaneous-associated lymphoid tissue is associated with the cutaneous region of the body, otherwise known as the skin. It contains T cells, monocytes, macrophages, and dendritic cells. In summary, the immune system is divided into the innate and adaptive portions. The innate immune system is nonspecific and generally responds the same way to every foreign entity, while the adaptive immune system is specific. Many different types of cells mediate the immune responses. Primary lymphoid organs are where the immune cells mature, while secondary lymphoid organs are where the immune cells are most active.